yeah, as Jason said, today I'm going to be talking about how we can engineer methyltransferases and then potentially use them as synthetic tools as well. So my talk today is mainly going to be based around alkylation reactions and how we can start to challenge nature's preference for methylation. Methylation is prevalent throughout nature, from epigenetic gene regulation to up and down regulation of neurotransmitters, and to being implicated in disease as well. So things like Parkinson's and oncology and many things in between. It is well known that the addition of a single methyl group to a compound can vastly change the chemical properties of said compound, with the pharmaceutical industry often utilizing the magic methyl effect to tune the pharmacokinetics of their APIs. And within nature, these methylation reactions are most often carried out by SAM-dependent methyltransferases. As it says in the name, these enzymes utilize SAM, or S-adenosyl-L-ethionine, as a cofactor. This cofactor, again, prevalent in nature, second only to ATP. And it is utilized as an electrophilic source of a methyl group to transfer to a range of different nucleophiles in nature. So we've got large biomacromolecules such as DNA and proteins, to smaller primary, secondary metabolites and other natural products as well. So prevalent throughout nature. Now, these enzymes are often found to be quite promiscuous, luckily not only towards their substrate scope, but also towards the cofactor as well. So you can vary this SAM cofactor either on the purine ring, on the amino acid chain to make isosteres, to make it more stable, or you can vary this transferable alkyl chain here, so we can extend it beyond the methyl to perform non-native alkylation reactions. Two examples I've shown here, rapamycin and rebecamycin. These are both pharmaceutically relevant compounds with their respective methyltransferase enzymes have been used to perform non-native alkylation reactions. Now, one of the enzymes I'm going to focus on today is CNMT, or cochlorin N-methyltransferase. This enzyme is involved in plant secondary metabolism towards the production of benzyl isoquinoline alkaloids. These compounds are based on this core tetrahydroisoquinoline structure. This is considered to be a privileged scaffold, in that it is used in the production of a number of different pharmaceutically relevant compounds, from muscle relaxants to antibiotics, anti-tumor agents, and many things in between as well. So very interesting enzyme. In 2018, the Microphil group solved the crystal structure of CNMT for the first time. We used this crystal structure to perform point mutations in the active site of the enzyme and figure out what residues were important to enzyme activity, and from this propose a catalytic mechanism. So we propose it goes via deprotonation of the substrate amine from a histidine residue in the active site that facilitates that SN2 nucleophilic attack onto the SAM cofactor, performing your methylation reaction. Now, we wanted to show that CNMT is not a one-trick pony, so to say, that it could potentially be used to structurally diversify a range of different tetrahydroisoquinolines. As I said, it's a privileged scaffold, so the more we can modify, the better. So as you can see in the top right here, I've shown how we used a range of readily available compounds that we can buy from, say, Sigma-Aldrich, highly commercially available variations at these aromatic methoxy and hydroxy residues. You can swap them, change them, take them away, add on some steric bulk, and the enzyme is generally quite well tolerant of this. Moving on to some other commercially available tetrahydroisoquinolines, we have increased steric bulk again. We have aromatic residues, hydrogen bonding residues, smaller methyl and ethyl groups. These are all generally quite well accepted by the enzyme. The second you start to branch out, get more steric bulk without compensating with aromaticity, or you put charged polar molecules in there, it sort of starts to destabilize this hydrophobic binding pocket. Moving on to something that was a little more tricky is a C4 substituted tetrahydroisoquinolines. Now, these aren't commercially available. These have to be synthesized in-house, which proved to be a lot more tricky than we originally thought. But as you can see, modification at this position is generally not quite well accepted by the enzyme. We see small modifications, methyls, dimethyls are okay. The second you start to build upon that, though, the enzyme activity goes down or is not active at all. So quite a broad range of modifications at many different positions, quite well supported by this enzyme. We wanted to take this one step further, however, and show that CNMT could also be promiscuous towards the cofactor as well. So as I mentioned, we can utilize SAM analogues in conjunction with many methyltransferases to perform these non-native alkylation reactions. And within the literature, these are mostly limited to the set that I've shown here. So we've got short to medium chain unsaturated arms on the R group here, so the transferable alcohol chain. And this is because these are considered to be doubly activated analogues in that they overcome the increased steric bulk in the active site by performing something called conjugative transition state stabilization. Now, there are ways we can overcome this, and we can diversify the SAM analogues that we can use. But this mainly comes from engineering the methyltransferase itself. So, for example, bump and hole mutagenesis, where you sort of carve out space in the active site around the increased sterics that you want to add. On the same vein, alanine screening, this is a bit more random. You mutate random points in the active site to a smaller alanine residue and see what effect this has on enzyme activity. Or there's a route we went down, which is structure-guided or rational mutagenesis, where you look at the amino acids that surround the area you want to add your bulk into, and then you see how you can further stabilize that and hopefully destabilize the natural cofactor at the same time. Now, these SAM analogues aren't trivial to make. They're a pain, they're very unstable, they degrade at the drop of a hat. I think SAM has a half-life, if I can remember, of about 100 minutes, so about 2 hours. And you can synthesize them in a couple of different ways, each have their pitfalls. So you can synthesize them synthetically from SAH or S-adenosyl homocysteine. This is basically just demethylated SAM. And the corroborating alkyl halide with some base in aqueous solution. Overnight, you tend to get yields from 10 to 60%, which then require separation as it produces both S and R epimers of the SAM analog. So with the S being the naturally occurring analog that is accepted by the enzyme, and the R actually being inhibitory to enzyme activity. So they require separation by chiral HPLC. Again, with the stability issues, this isn't great as the compounds tend to degrade when you're purifying them. And to combat this production of S and R enantiomers, you can do it enzymatically. You still require the synthesis of your methionine analog. However, you can use methionine adenosyl-transferases with ATP or halogenases to do this enzymatically. Again, this will need purification from your reaction mixture before you can use it, but at least you only have the one sulfonium epimer to contend with. But these tend to be done on small scales, and both fall into the pit of not being membrane permeable. So these SAM analogues can't permeate the cell membrane, so are largely limited to smaller scale in vitro applications. And we wanted to push past this and be able to determine a mechanism where we can apply a SAM analog in vivo produced within the cell itself. So there's no stability issues, no storage problems, and no cell permeability issues either. So we move towards this first and only known naturally occurring SAM analog, Carboxy-SAM. This was discovered in 2013 and is produced from a carboxy-SAM synthase enzyme, CMOA, using SAM that is present and exogenous within the cell. Now, the carboxy-SAM is then utilized by our carboxy-methyltransferase, CMOB, to transfer this carboxy-methyl functionality to the wobble position of a tRNA substrate. We wanted to hijack this process and combine it with an engineered methyltransferase and our core tetrahydroisoquinoline substrate for the diversification of this privileged scaffold. So in order to do this, as I said earlier, we looked towards rational structure-guided mutagenesis, and that we looked at the active site of CNMT and we compared it to the active site of our carboxy-SAM methyltransferase, CMOB. This enzyme has naturally evolved to have a 500-fold increased affinity for carboxy-SAM over SAM. And looking at the active site, you can see why. So there's a range of positively charged arginine, lysine residues within the general vicinity of that carboxy-methyl arm, with the arginine directly stabilizing it. Conversely, in CNMT, we see almost the exact opposite. So you see a lot of negatively charged glutamic acid residues that are primed for mutating to try and mimic this sort of interaction here. That's just what we did. So we found that in particular, these Y81 residue mutations gave us a significantly decreased activity with SAM and an increased activity with carboxy-SAM, with this Y81R mutant being the best. You see this a little bit better in these time course assays. So the stark difference after 1 minute of reactivity, the wild type is almost completely finished its reaction, whereas with the mutant and SAM, we get less than 1% after a minute. Looking after an hour, we get a decreased drop in 60% activity with the wild type, with the mutants compared to the wild type, an increase of 15% with carboxy-SAM. This is nowhere near perfect, we realize this, but it is a one single point mutation that has enabled us to direct towards more orthogonality. Now, in order to get a system in which we can make carboxy-SAM and do carboxymethylation in situ in one pot, we first had to optimize the production of carboxy-SAM from SAM using our carboxy-SAM synthase. This enzyme utilizes prephenate as a carboxy donor, which is in turn made from chorismate. This can be done enzymatically using a chorismate mutase, but it can also undergo spontaneous rearrangement. We found that after 5 hours in our assay conditions, these processes were essentially the same, or the same rate. So we negated the use of chorismate mutase and ended up with reaction conditions after 5 hours that gave us 80% conversion to carboxy-SAM. Using these conditions going forward, combining them with CNMT and heliamine, so this tetrahydroisoquinamine scaffold, we were able to find conditions where we could get 70% carboxymethylation and only 16% methylation. So after 5 hours, when the methyltransferase is introduced, we expect there to be 80% conversion to carboxy-SAM, so 0.1 millimolar SAM left, 0.1 millimolar substrate. You should get 100% methylation if the enzyme is still preferring its natural cofactor, which it's not here. So orthogonality has been achieved in this case. And we wanted to see if we could easily apply this to another enzyme. So we picked COMT, or catechol-O-methyltransferase. This methyltransferase is involved in the degradation of catecholamine neurotransmitters. It's also implicated in Parkinson's disease. And looking at the active site, it's a lot larger, a lot more flexible and nearer the surface of the enzyme compared to CNMT. So we expected it to be a lot more promiscuous towards its cofactor, hopefully. We also identified this M40 residue, which was within 3 angstroms of this transferable alkyl moiety, so the methyl and SAM, carboxymethyl and carboxy-SAM. So this was the first protocol for mutagenesis for us. Now, using the wild type on its own and our assay conditions, we were able to achieve near 60% carboxymethylation, 12% methylation. Again, this is not perfect, but using a wild type enzyme, I think it's pretty impressive, considering that this enzyme has evolved over millennia to accept SAM as its natural cofactor. Using a mutant that is known to the group previously for the improvement of the regioselectivity of the COMT enzyme, so making it highly selective for the meta position over the para position, we were able to see this replicated with carboxy-SAM, so we get increased regioselectivity. Introducing our M40 mutants, we found that after 17 hours, we didn't see a decrease in SAM activity, which we don't expect after 17 hours, but we do see an increase in carboxy-SAM activity, which is nice to see. Combining our M40A mutant, which we thought was going to be the best one going forward, and the Y200L mutant, we were able to completely mimic that change or increase in regioselectivity. So through 2 single point mutants, we are able to increase cofactor selectivity for a new novel cofactor and almost change regioselectivity completely. Another good example of this is a different substrate for COMT. So this is a nitrocatechol. Again, similar results as last time with the wild type enzyme. And this time we see with the M40A mutant, we get a complete swap in the regioselectivity. So now we're looking at way more para-methylation than carboxymethylation than we are meta-carboxymethylation. Again, when you combine this with the Y200L mutant, we can completely swap that back around again. So we can go meta, para, and more carboxy-SAM selective, just again through 2 single point mutations. Taking this one step further, there are examples in the literature, though they are quite rare, where a methyltransferase has been utilized with a prochiral substrate in order to form a new chiral center. Before this, there were no examples, however, of a chiral center being directly transferred from the cofactor itself to a substrate. So in order to mediate this, we envision the creation of this new novel cofactor, Carboxy-S-adenosyl-L-ethionine. So you've got this additional methyl group here creating a chiral center. We made this synthetically using SAH and bromopropionic acid, as well as enzymatically from ethionine and the carboxy-SAM synthase. And in all circumstances via chiral HPLC, as compared to a synthetic standard, we only ever saw the R isomer. We hypothesized from the CMOA active site that only the S at the sulfonium center and S at the carboxymethyl center is formed by the CMOA enzyme. This then undergoes inversion of configuration when attacked by the substrate to give you the R product. We envision this goes because of this F133 residue in close proximity to that potential methyl group that we have here. So we expect it will be out pointing towards you, as we've shown here, instead of clashing with this residue. So you get deprotonation from one face, subsequent decarboxylation supported by this R199 residue, and then you get carboxylation from the V face, giving you your S enantiomer. Going on to the CNMT active site and seeing whether CNMT itself would utilize S or R carboxy-SAE. We did some docking studies. So in green here, you can see the docked SAM sits directly over the crystallized SAM in the crystal structure. So it's a proof of concept that our docking studies are actually working okay. And when you add in SR carboxy-S-adenosyl, so S at the center, R at this center, you can see that there's a shift out of the active site. So it's shifting away from the site of nucleophilic attack with this methyl group directly in the way, blocking that attack anyway. With the SS carboxy-S-AE, we see that it sits exactly where we want it to sit, with that methyl group pointing out of the way of the activity, primed for nucleophilic attack. So we estimate that CMOA would only form SS carboxy-S-AE, and consequently CNMT will only accept SS carboxy-S-AE to give you your R product. Moving on to what the lab is currently working on at the minute from this work. So as I said before, the whole purpose of the utilization of carboxy-SAM and the production in situ of our cofactor is that we could move this into a cell and overcome permeability and stability issues. And that's exactly what we're trying to do. So we have CMOA that's produced within the cell, forms carboxy-SAM from our SAM that's exogenous, and then we have our methyltransferase made within the same E. coli cell. You supply exogenously your tetrahydroisoquinoline scaffold, and you get carboxymethylation in situ. Again, this is a work in progress at the minute, not an idealized circumstance with results at the moment, but it's in progress. So certain ways that we can help mediate this is to try and increase carboxy-SAM concentration within the cell. We can do this by engineering metabolic pathways, so trying to knock out some of the potential pathways that utilize these carboxy donors, sort of increase carboxy-SAM concentration in the cell. We can also vary and stagger protein production so that we have CMOA kicking around for a long time before we introduce the methyltransferase, trying to mimic those in vitro conditions that we developed. Moving on from this, which I think would be quite cool, is the production not only of CMOA within the cell, methyltransferase within the cell, but also a substrate. So if we utilize a protein methyltransferase, we can potentially form in vivo protein carboxymethylations as well without any exogenous supply of reagents or substrates. It's all made within the cell itself. You just grow it up, you isolate it at the end, or you do your analysis. So this acts as a handle for further derivatization, bioconjugation, amide coupling, photoredox chemistry, you name it. Yeah, it's a lot more useful than the single methyl group anyway. And we can apply this to a potential diverse range of methyltransferases. So they're prevalent throughout nature. Yeah, one single active site mutant could potentially give us activity with carboxy-SAM and a range of different substrates as well. Yeah, if you're interested, these are the two reference publications from the work that I have talked to you about today. And with that, I would like to thank Professor Jason Mickelfield for all his help over the past 3 years. It's been an absolute pleasure working at the University of Manchester in the MIB. Thank you to BBSRC and GSK as well for funding this project and the rest of the Mickelfield group. And thank you for your listening and the opportunity. I'll take any questions.
Thanks, Abby, for a wonderful talk, opening up the world for biocatalytic alkylation. Very exciting. Do we have any questions in the Q&A tab, Anthony?
No questions coming yet, but maybe I can start then. So yeah, thanks very much, Abby. That was a fantastic talk. So it seems like basically you can adapt the cofactor preference relatively easily with a couple of mutations. Do you guys plan to try and really specialize that enzyme to specialize that conversion through evolution, for example, to try and get highly selective enzymes for carboxymethyl? So
it's very possible, yes. Something like that is very possible, very easy, especially with the automation processes and the equipment that we have in the MIB here. I can definitely foresee it being a simple process. However, this research at the minute is more based on a proof of concept that we could potentially do this with a vast range of methyltransferases rather than specializing one individual methyltransferase to be perfect at carboxymethylating, for example, at this moment in time. That's not to say that, yeah, it's not a great idea for the future.
Okay, so we have, questions are starting to fly in now. So a question from Tristan Durand. If you wanted to engineer the free carbon transferase system into whole cells, I'd be worried about the toxicity of the thionine starting material to the cell. Any ideas how you might circumvent that?
Yeah, this is something that we have thought about. The carboxy-ethylation was potentially not so much in order to put it into a cell. We mainly focused on carboxymethylation for that, for the reasons that you've highlighted. We mainly were trying to probe the stereospecificity of the enzyme in that circumstance and show that it's possible that you can perform these reactions in vitro stereospecifically. Yeah, in vivo would take a lot more due care and consideration on that front, especially with not only ethionine, but other methionine analogues as well.
Okay, so we have a question from Matt. Does Carboxy-SAM display similar stability issues as SAM? And any ways to counter this instability in vitro?
So yeah, Carboxy-SAM, especially in my hands anyway, has a pseudo-similar half-life to SAM. So we see very similar degradation products. So you see the methionine, the methylthioadenosine and the homocysteine lactone, or homoserine lactone, sorry. We don't see decarboxylation all that much. And if we do, it's very minor. However, in vitro, we don't expect this to be an issue, as we expect the concentration of Carboxy-SAM to be that high and the methyltransferase concentration to be high as well because of the production within the cell. So we condition the E. coli cell to produce CNMT and CMOA almost exclusively. So I don't think that the stability issues in vitro for Carboxy-SAM would be an issue in the start. At least it hasn't been in the work that we've done so far. But there is the possibility of work with isosteres and things like that that overcome these cyclization issues. So we can put isosteres on the amino acid chain that stops that in vitro, that cyclization from occurring. So yeah, so it's a possibility. I think isosteres would probably be the way to go forward on that.